Hi, my name is Dung.
And I'm Alexandra Ostezan. We are PhD candidates in plant breeding and genetics at the University of Georgia.
Soybean seed nematode, or SCN, is small plant parasitic roundworm that attacks soybean roots. The nematode ability to report in China in 1899 and in Japan in 1915. It was firstly reported in North America in North Carolina in 1954 and spread throughout the soybean producing states and became the #1 enemy for soybean growers. Of all soybean affliction, SCN caused 1/3 of all soybean yield loss, accounting for over $1.2 billion annually in the United States alone.
Soybean farmers fertilize their crops, and this helps the plants grow well. But this also means that there are more nutrients which the nematodes can ingest through the plant. Juvenile SCN travel through soil and use their stylets to penetrate the roots, establishing a permanent feeding site and robbing nutrients from the soybean plant. When soybeans are infected with SCN, they can show above-ground symptoms like stunting and yellowing leaves. However, these symptoms can be confused with other nutrient deficiency afflictions. Sometimes, soybean plants can be heavily infested with SCN without displaying visual symptoms. The SCN life cycle has about 28 days, and a single female can contain up to 600 eggs. Susceptible plants can have hundreds of females, and during a growing season, 3 to 4 life cycles of SCN occur.
Although SCN doesn't travel great distance alone, they can be transported from field to field through farm equipment, water, soil, winds, and animals. They can stay in the soil for up to 10 years, so growers are recommended to send the soil samples to diagnostic labs to detect SCN present in their fields.
To beat SCN, soybean breeders have screened germplasm from around the world for resistance. A standout resistance source was identified and used almost exclusively for more than 20 years. This plant provided a great protection for soybean plants from SCN attack. It leads to the slow degeneration of a feeding cell, preventing the SCN from absorbing more nutrients. However, due to natural selection and using the same resistance source for a long period of time, SCN has evolved to adapt and overcome the resistance. Gradually, they have been able to feed and reproduce in larger numbers.
Now we are working to deploy new resistance to SCN in soybean plants available to farmers. One solution is a pyramid of various resistance sources. This means using multiple resistant genes in the same plant simultaneously. Pyramiding is useful in reducing the effects of SCN population on plants and also slow down the nematode evolution of beating the resistance. Despite the nematode ability to overcome the commonly used resistance, utilizing the genes from the plant in conjunction with other resistant genes or QTL leads to enhanced resistance of the plant. The genes have different functions, ranging from localized cell death in the plant surrounding the nematode to specific degradation of feeding site.
Current known genes provide the resistance utilized by producers today. Looking forward to the resistance of the future, soybean breeding and research programs are searching for new, undiscovered resistance genes. These can come from various sources, including wild, unadapted soybeans and accessions. A common method of discovering these genes is through biparental population, creating a new population of plants with a resistant and a susceptible parent. After a population is sufficiently inbred, the plants are inoculated with nematodes to test for resistance, and then genetic mapping work is done with the end goal of pinpointing the gene controlling the resistance and developing a molecular marker to easily determine if the resistance source is present.
Not only can the plant be tested for resistance with phenotypic assays, where the plants are inoculated with nematodes and resistance and susceptibility are quantified, but they can also be tested through marker assisted selection. Molecular markers are incredible tools for quickly testing to differentiate between resistant and susceptible DNA. We do a test on the DNA and then read the result in a machine that provides simple visualization of the scores. Here we can determine if the plant will be resistant or susceptible.
With these tests, we are working to provide soybean producers with more options for plants with the ability to win the nutrient battle.